Dear students, in the last lecture we had studied cell cycle and mitosis, a very important topic which is affiliated to our earlier studied topic, which is of mitosis, is mitotic apparatus. Now the mitotic cells, that means the cells which are undergoing mitosis have two apparatuses. One is chromatic apparatus and achromatic apparatus. In the chromatic apparatus, we take chromosomes and nucleolus, whereas in achromatic apparatus, we generally take centrioles, the spindle fibers, and kinetochoric fibers. Now come to the components of mitotic apparatus. In the mitotic apparatus, we take spindle fibers. The spindle fibers are nothing but they are aggregations of microtubules which are attached to centromeres of the chromosome, is known to most of you. They extend from pole to the centromeres. And when they shortens, the chromosomes are pulled apart towards the poles, whereas their elongation affects the movement of chromosome towards the pole. In fact, they get pushed towards the poles through the elongation. Now the spindle fibers, when esters are present around the centrioles, such spindle fibers are called as estral, and whereas anestral spindle fibers are without centrioles and esters, as depicted in higher plants and protists. The spindle fibers are of two kinds: kinetochoric spindle fibers, which extends from centromeres to poles. When they shorten during cell division, they pulls the daughter chromosome apart towards the poles. Another kind of spindle fibers are present which are called as continuous spindle fibers. They extend from pole to pole without attaching to the centromeres. And during cell division, they elongate and pushes the spindle poles apart, separating the chromosomes. This is their worthfulness. The interchromosomal spindle fibers are also present. They are continuous spindle fibers but are present between the chromosomes, and when they polymerizes, they pushes the chromosomes apart to the poles. Then come to the structure and chemical composition of the spindle fibers. The spindle fibers, they are made up of microtubules, and microtubules, as I taught you in regular classes, they are discovered by D. Robertis, and they are absent in certain cells like that of amoeba, RBCs, slime molds. Microtubules are actually unbranched hollow cylinders which are made up of 13 subunits which are called as protofilaments. As you can view it in the slide, that these 13 protofilaments, they are arranged at the periphery, and they are around 250 angstrom in diameter. Chemically, they are made up of tubulin proteins. A tubulin is a dimer of two polypeptide chains, alpha and beta tubulin. And they have strong affinity for magnesium ions. When the alpha and beta tubulin, they nucleate, or they undergo the process of nucleation, that means formation of around 340 angstrom aggregates of tubulin. Thereafter, growth happens on this nucleus by addition of dimeric tubulin. The tubulin polymerizes spontaneously into 13 protofilaments, and in this growth, calcium ions plays important role. When the two polypeptides of tubulin, the alpha and beta tubulin, they just find around 37 degrees centigrade of temperature with guanosine triphosphate and ample amount of magnesium. They get converted into protofilaments, and these protofilaments afterwards form microtubules. Now function, as I had mentioned earlier, the movement of chromatids to the poles during the anaphase. And in the earlier lecture, in the last lecture of mitosis, I had mentioned colchicine, which is an alkaloid from Liliaceae family. It binds to tubulin protein, 1 mole colchicine to 1 mole tubulin, so that mitosis stops at metaphase. This is the reason why colchicine arrests mitosis at the metaphase state, as spindle is broken down and polyploidy happens due to this. Now, what are esters? At the prophase, each centriole divides into two. Each gives off estrol rays made up of microtubules. The centrioles or centrosomes, they were discovered by Van Banden and have 9+0 microtubular arrangement. Centriole is actually a cellular organelle of microtubular organization, and it is found only in animals, and during cell division they migrate to the opposite poles, ensuring the separation of the replicated. The area near the centriole is very much clear and is devoid of any kind of organelle, and this clear area is termed as centrosphere. Centrioles occurs in all animals excepting higher plants, prokaryotes, diatoms, and yeast. In higher plants, they have microtubular organizing centers instead. If we go to the structure of the centriole, it is a cylindrical structure open at both ends, and its wall is made up of 9 triplets of microtubules, equally spaced and filled with some material, and the triplets of the microtubules, they form a cartwheel like structure, and these triplets, they are linked by linkers. As far as intracentriolar structure is concerned, it consists of 9 radiating spokes from hub of around 25 angstrom, and chemically the centrioles, they are made up of tubulin, some amount of ATPase enzyme is also present, some nucleic acid and lipids. Now just see this structure. This is the cartwheel structure of centriole, and there are you can view the triplets of microtubules, and these triplets, they are made up of 13 protofilaments, and 9 triplets, they are arranged at the periphery, and 9 radiating spokes, they arise from the central hub. And these triplets, they are linked with linkers, and the protofilaments, 13 protofilaments, they are made up of microtubules, which are polymerized form of alpha and beta tubulin. So this 9-spoke cartwheel structure is of centriole. It is the characteristic of centriole. Now, anaphasic movement. As the spindles, they are very important as far as anaphasic movements of the chromosome is concerned. It is worthy to mention the role of microtubules in the anaphasic movements of the chromosome. Now, dynamic equilibrium hypothesis was given by Now, and he mentioned disassembly of microtubules responsible for dragging of chromosomes during the anaphase to the opposite poles. And the rate of movement of chromosomes to the poles is directly proportional to the rate of microtubular disassembly. uh, or depolymerization. In fact, the spindles are attached to kinetochores, and when the tubulin protein depolymerizes at the poles, it shortens the spindle fibers, dragging the chromosomal chromatids towards the poles. The monomers are removed at the pole, reaches the interchromosomal spindle fibers, and assemble there. And when their length increases, it pushes the chromosomes apart also. You can understand this from this diagram, which is illustrated, drawn there at the bottom, that this is the metaphasic chromosome. I'm talking about the diagram on the left of your screen, that this is metaphasic chromosome, and at the middle you can see the centromeres, and to the centromeres we have attached spindle fibers, and you know that spindle fibers, they are made up of microtubules. And these spindle fibers, or kinetochoric spindle fibers, they start their disassembly or depolymerization. When they depolymerizes, they exert a force, certain force, which is sufficient enough to break these daughter chromatids from the point of centromeres. When they break, the depolymerized spindle fibers, they just migrate to the place or space between the two chromatids, daughter chromatids, and they start polymerizing there. This activity is ATP driven, and when they polymerize between two chromatids, it pushes the two chromatids towards the pole, whereas the depolymerization in the first diagram pulls the chromatids towards the opposite poles. Another hypothesis which was given by McIntosh was sliding hypothesis. It is something analogous to muscle contraction. And you can view at the bottom of this slide that the chromosome is attached, chromatid is attached to the spindle fibers, kinetochoric spindle fibers, and what happens that these kinetochoric spindle microtubules slide along the microtubules of pole to pole spindle fibers. Pole to pole spindle fibers are those fibers which are not attached to the chromosomes. And for intermicrotubular bridges, dynein-like arms have been noted in the spindles. These dynein-like arms work against the actin filaments of the spindle fibers, which are present in the spindle fibers attached to the kinetochores. The force is generated by myosin-actin interaction rather than by tubulin-tubulin interaction. This is also ATP driven. So students, this is all in this lecture. We had studied mitotic apparatus. Thank you so much.